All right, so if you go to the first page of your Part 2 notes for Chapter 2, you're going to see the following questions provided. I'm going to leave this screen up while you are recording your notes because it's not going to be provided on the PowerPoint in this order. So the first thing that you were to answer is, what is a chemical reaction? So in a chemical reaction, you're going to have a substance either being created, broken down, or rearranged. You're going to have two primary parts of a reaction. That is the reactants and the products. The reactants are actually what is being changed, and then your product is going to be the new substance that you receive from that reaction. If you look at what are the types of reactions, I have three listed: synthesis, decomposition, and exchange. So RXN is my abbreviation for a reaction. I do apologize. I have to get out of the habit of using those things when I'm making notes for you all. So let's look at synthesis. Synthesis is the joining of components that were previously separated. Usually in a synthesis, you're going to have a new product that is created. So in our example, I have A plus B equals AB. So that is your synthesis reaction. In a decomposition, it's going to be the opposite. So rather than something being produced, you're going to have something being broken down into its original components. So decomposition example was AB equals A plus B. So at the conclusion of this reaction, the A and the B were separated back to just being AB instead of AB combined. Finally, the third example is your exchange. In an exchange reaction, you're going to have both a synthesis and a decomposition that is going to happen. Chemical bonds are both broken and formed in exchange reactions. So if you look at the examples just under 3, you have A plus BC. So the result, or our product, is going to be AB plus C. So if you look at the original and then look at the new product, you see that the A and the B were joined after B and C were separated. The other example gives you where you have two components that are being switched. So I have I have AB plus CD, which equals AD plus BC. So in this one, the B and the C were separated. Were combined, and the A and the B and the A and the D were put together. I know I just blah. But basically what I wanted you to see is that the letters switch places. In chemical reactions, you're going to have the presence of two types of energy: kinetic and potential. Kinetic is always going to be the energy that is in motion. So when the reaction is occurring, when those molecules are moving, that's a kinetic type of energy present. But if it's not necessarily an activated reaction yet, that's when you're going to have potential. So it's in its position, it's ready to go, but because it's not currently in motion, it has the potential to, elude or release that ATP energy that's probably being used. Both of these energy types are going to be interchangeable. All right. All right, so the next part of your notes is talking about an inorganic substance. Most of the chemicals in the body are going to be compounds, either inorganic or organic. Specifically, what you need to know about inorganic compounds is that they usually don't have the presence of carbon. There are two examples that are an exception to this rule. That would be carbon monoxide, which is CO, and carbon dioxide, which is CO2. Even though the carbon is present, they are still considered inorganic compounds. Usually you're going to have water, salts, acids that will be present within the body that are going to be your examples of inorganic compounds. Organic compounds, which if you go to your what page is this? The back of the first page. I'll pause and let you flip to that one. All right, your organic compounds are going to have carbon present. They're always going to be in a covalent bond. As we continue through the slides, we'll look at examples of both. Okay. Next thing is acids, bases, and salts. So acids are going to be obviously more acidic, but what's causing the acidity in them is the presence of the hydrogen ions. All three bases, acids, and salts will dissociate in ions, which means they're going to dissolve in water. Because acids release their hydrogen ions in that water solution, that causes them to be strong acids and also be more highly reactive toward hydrogen. Some examples of your highly reactive or strong acids will be hydrochloro, well, HCl. I can't remember how to say it right at the moment. But that's going to be produced by your stomach. It aids in digestion, and then that particular acid is also going to kill off the microbes that are within the body. Another type of acid that wouldn't necessarily be one that you produce, but one that's very beneficial for you, is an acetic acid. Vinegar is an example of acetic acid. A lot of people will ingest vinegar to stop bacteria growth or fungus growth within the body. What is another one? Well, those are the main ones, but there are a lot of health benefits to vinegar, again, which is your acetic acid. Acids and bases will react in the body to create salts. Some of those important salts are going to be electrolytes, which help with the production of a nerve or muscle impulse. So real quick, bases. Both, again, ionize in water, but rather than releasing hydrogen, bases are going to release hydroxyl ions, which is going to be represented as an OH with a negative charge. It's also what helps water form. It removes the acidity in a substance when it's placed there. All hydroxyl ions are weak unless they have released all of their hydroxyl ions. So very similar to acid, where the stronger acids are going to release all of their hydrogen ions, stronger bases are going to release all of their hydroxyl ions. A popular and very useful base would be bicarbonate, or some type of bicarbonate. Usually sodium bicarbonate is going to be the one that's found in your antacids to relieve your heartburn. Alka-Seltzer is another example. These are going to potentially be weak bases, and their role is to reduce the amount of acidity that's present in a substance. Okay, what are some properties of water? All right, the ones that you need to record is that water is the most important inorganic compound in a living system. It's going to be the medium for all chemical reactions, and what I mean by that, it's going to be a tool that those reactions are using in order for that to occur. It's also a cushion or a type of lubricant within the body. We briefly talked about the polarity of water being that it has a negative charge side and a positive charge side. So the sharing of their electrons are unequal. This actually makes it an excellent tool for breaking down your ionic your ions. It gives water its molecule cohesion, and it allows it to dissipate heat because it has a moderate change in temperature. Water is also our universal solvent. It's talking about polarity and the covalent bonds with the hydrophilic versus hydrophobic. I'll get back to this when we talk about cell membranes and some of the components that is within that. Water is able to dissolve or suspend many substances. So that's just a few of the items you may want to record for properties of water. All right, going back to our Cornell note sheet. We're looking at what is the pH scale. So as you can see on the slide, it's going to be a range from 0 to 14. Those that are higher on the number are going to be more basic. Those that are lower are going to be more acidic. 7 is going to be neutral. So I don't think I said that right in the right order. So let me restate. The higher numbers above 7 are going to be basic. The lower numbers close going towards 0 are going to be the most acidic. I'm not sure if I said that at the first one or not. Let's see. To make sure that I'm getting everything from this one. The only other thing was the concentration of the ions that are present is what determines which direction it's going to go. So the concentration of ions at each pH is 10 times the value of the next, either above or below. So if we're starting at neutral, an example of a neutral substance is going to be water. 7.2, 7.4 has an excellent acidity for the body. And then as you start to go down from 7 to 6, that's when you start to get into your acids. And if you look at the chart with the blue and the white, it gives you the number of moles per liter that are present, both in your hydrogen ions and your hydroxyl ions that are present. Okay. All right. The buffering system of the body. So your body fluids are going to have varying pH, but they have a certain range that they have to maintain. For example, your gastric juices have to be between 1.2 to 3.0. Saliva has to be 6.35 to 6.85. Your bile is 7.6 to 8.6, and your pH of blood is going to be 7.35 to 7.45. Buffers convert the strong acids to weak ones, which contribute to fewer hydrogen ions being released, and you have a smaller effect or a less effect on your pH. The system of molecules and ions that prevent this drastic change in your pH is what the role of the buffers is. It's a stabilizing of your pH in any solution. Let's see. All right, so we briefly talked about organic compounds. We know that they're always going to contain carbon. In most cases, they're going to have hydrogen as well. They usually are kept together by a type of covalent bond. They're going to be larger molecules with a complex function. Organic compounds compose about 40% of your body mass, and some examples are carbohydrates, lipids, proteins, DNA and RNA, and ATP. And we'll look at each of these briefly. Excuse me. So let's look at our carbohydrates. Carbohydrates are a diverse group of substances. As I previously mentioned, will contain carbon and hydrogen, and also in most cases they're going to have oxygen present as well. It is a water carbon because it's hydrated a lot. Some examples of carbohydrates will be your sugars, starches, the fibers that you receive from fruits and grains. Some vegetables have carbohydrates, as well as some milk products. There's usually going to be a 6-carbon atom chain and 6 water molecules present. Main function of carbohydrates is that they produce a source of energy for the body, which is your adenosine triphosphate, or ATP. Diversity of carbohydrates. This slide is just basically giving you a breakdown of the carbohydrates and how they're classified. So your monosaccharides are going to have 1 sugar. They'll usually be a glucose or fructose. Your disaccharides will have 2 sugars. They can be in the form of glucose and fructose, which will give you sucrose. Polysaccharides are going to be larger. They're going to have up to 100 types of monosaccharides that are combined together. Usually they're going to be associated in a format which produces the glycogen. All right, lipids are going to be our fats. Again, it's also going to be composed of carbon, hydrogen, and oxygen. Types of lipids would include phospholipids and steroids. Lipids are usually going to be hydrophobic in that they do not like water. So they would be classified as water-fearing. Okay. The next thing on here, main types of carbohydrates, lipids, triglycerides. All right, so with your triglycerides, you want to know that it's the main component of the body fat. It's located in blood, and it's this place where we store our energy. It's a very concentrated form of energy. Our bodies store triglycerides in fat cells. So if we eat extra food and we're not able to burn off the calories, the excess ATP or the excess fat that's left over will be stored in our triglycerides. All right, so here's a picture of the structure. It's not something you're going to have to identify for me. Just kind of giving you an overview of how they look. Next type of fat are your phospholipids. The composition of the phospholipid molecule is that it has a polar head and two nonpolar fatty acid tails. These are the main constituents of your cell membrane, plasma membrane, depending on which book that you read. The head is going to be water loving. So it's going to be semi-permeable. It allows certain molecules and substances to pass through, whereas your fatty acid tails are going to be more water phobic or water fearing in that they don't allow water molecules to pass through. In your cell membrane, you're usually going to have some type of protein located within it or another type of way for molecules to pass through. When we get into chapter 3, we'll look at that a little more in detail. All right, so the circle here is going to be your polar head, and then these are going to be your nonpolar tails. In the lower diagram, it's giving you an idea of what your cell membrane looks like, with the polar heads being semi-permeable, and then you having your fatty acid tails in the middle. All right, so let's look at steroids. It's letting you know that it's a 4-ring of carbon atoms joined together. Some common steroids that the body produces would be those associated with your sex hormones, bile salts, vitamins, and cholesterols. You do have some of these materials also located within the body. Let's see. Estrogen, progesterone, testosterone would fall under the sex hormone category. Most of these are going to be produced from your adrenal gland. And in the adrenal cortex is where you're going to get those different types of hormones produced. So if you have a corticosteroid, that's going to alleviate swelling within the body. Your antibiotic steroids are the ones that you can take over the counter that build muscle. They're referred to as your constructive metabolism type of steroids. Proteins have a critical role in the body because they provide the cells with their structure and allow them to, carry out their functions in the body to regulate our tissues and our organs. So without our proteins, our tissues and organs wouldn't function properly. They're going to contain carbon, hydrogen, and oxygen. Most of your proteins are going to also have nitrogen. There are 20 different combinations of amino acids that support these proteins. We looked at a list of them when we were going through our transcription translation. Let's see. Then you have your levels of structural organization. This is not something that you're going to need to know for your test, but I just wanted to give you a brief overview of the levels. You have your primary, secondary, and tertiary. And the shape of those particular proteins from their amino acids, it's what influences their ability to form certain types of bonds. All right, so with our amino acid structure, know that the amino acids are going to be your building blocks for proteins. Most of them or some of them are not going to be produced in the body. Those amino acids you're going to gain from your diet. And it's important that you have a diet that provides you with those amino acids in order to support the function of the proteins in the body. Loss of amino acids can result in muscle loss. It can prevent the tissue repair or organ repair within the body as well. With the amino acids, those that are considered non-essential are made or produced by your body. And the ones that are essential are the ones that I spoke of briefly about. You're needing to receive those from what you consume. And this one is just giving you a slight diagram of the different types of amino acids and how they're formed. If you look at some of the names, these were some of the ones that were listed on our amino acid chart that we used. Okay. So looking at the structural levels of amino acids, the primary level of amino acids is going to be some of the ones that we looked at on our chart with our transcription translation sheet. Secondary are going to be those that are going to be present in your DNA because they're usually double helixed. And tertiary are going to be larger molecules located within the body. You have your polypeptide chains. Just going to go through DNA structure and RNA structure briefly. We kind of talked about it in class. Huge molecules, they're going to be larger, so they're going to be the second tertiary level. They will contain carbon, hydrogen, oxygen, and nitrogen, and usually have a phosphorus as a backbone. Each gene in our body has genetic material that is the pieces of our DNA, and it controls the synthesis of protein. A molecule of DNA has a chain of nucleotides or nitrogen bases. The four we spoke of were A, adenine, G, guanine, T, thymine, and C, cytosine. The structure itself, here you have your phosphate group. This is your sugar, and then each one of these would be your nitrogen bases. And then we spoke about adenine always pairs with thymine when it's in DNA. Guanine always pairs with cytosine, regardless of if it's in DNA or in RNA. Slight differences from DNA to RNA is that DNA is single-stranded rather than double-stranded. It has a ribose sugar instead of the deoxyribose sugar, and that uracil replaces the thymine. There are 3 types of RNA located within the cells, and each one has a specific function. You have your messenger RNA, ribosomal RNA, and transfer RNA. And then finally, the only other thing we had listed in our notes was our adenosine triphosphate, or ATP. It is the energy currency for the body. So every reaction, every process that occurs uses ATP in some form or fashion. It does consist of 3 phosphate groups that are attached to a 5-carbon sugar, which is usually going to be your ribose. And then this is the structure here located below. All right, so that is the end of our Chapter 2.2 presentation. So the next time I see you all, we're going to go through Chapter 3. My goal is to get that one completed in one setting because I'm going to have to use 2 days to do Chapter 4. All right, enjoy the rest of your weekend.